I'm Anne Rancourt, reporting for HIV.gov in Boston at the Conference on Retroviruses and Opportunistic Infections. And I'm here today with my colleague, Dr. Carl Dieffenbach, Director of the Division of AIDS at the National Institute of Allergy and Infectious Diseases, part of the National Institutes of Health. Carl, thanks for joining me.
Always a pleasure.
Today's the last day of CROI, so we're wrapping up what we've heard today in the scientific sessions and panels that have been going on here. And before we break it down, I want to invite you to share any questions or comments that you might have with us. We'd love to hear where you're watching from, but also use this opportunity to ask us anything that you might like. So, Carl, today we heard about an interesting study about drug-drug interactions between a contraceptive vaginal ring and efavirenz, which is one of the most widely used forms of antiretroviral therapy. Tell us a little bit about that.
So let's back up and start from the concept of drug-drug interactions. People living with HIV and on antiretrovirals are probably taking other medications. So one of the areas we're very careful with is, do these drugs interact with each other and change the level of the drug that would be in the body? So for example, a drug interaction could make a drug clear faster, so the drug was not present in the body, so you wouldn't have the protective effect, or it could vastly increase the amount of drug in the body, causing potential safety problems. So what we saw today was the results of a pharmacology study, which means the drugs were given, and then physicians took very careful measurements of the levels of the drugs in the bloodstream. And what they were able to see is that there's a vaginal ring that releases contraception into the tissue, caused, when given with efavirenz, this very commonly prescribed antiretroviral, caused an 80% decrease in the level of the contraception in the bloodstream. That's a significant change, to a point where the lead investigator on the study said she would be very concerned if she were an HIV positive woman using a ring and being on efavirenz. So I think it's an important thing to let women know about, and so that they can adjust their choice of contraception, because family planning, as we've talked about at this conference, is a very important part of making sure we have healthy family, healthy babies, and an HIV-free generation.
And so for any HIV positive women who might be watching at home, this is something that they should talk to their doctor about and look into?
Absolutely. You know, particularly if you are currently using a ring-based contraception, talk to your doctor about the drugs that your antiretrovirals that you're on, so that you have the opportunity to make sure that you are doing what you think you're doing by taking your medications.
And so on this topic of drug-drug interactions, I mean, hormonal contraceptives are very widely used by women, many of whom may be living with HIV and also on antiretroviral therapy, hopefully. What other research is going on to look into this issue to make sure that women don't fall into this?
Well, I think it's very important that we think about all the different drugs that people take. So, for example, we've talked a lot at this conference about tuberculosis. So many of the TB drugs interact with the HIV drugs as well, and we've worked very hard to figure out how to dose the TB drugs with the HIV drugs so that both are done safely and effectively. So that's an example. And as we go forward and think about contraception with prevention and contraception in the situation of HIV therapy, there are all these drug-drug interactions that we need to work out. We are dealing with polypharmacy here. We need to make sure the polypharmacy is safe and effective.
And so, Carl, you've spoken a lot about your desire to create a product that delivers for women both HIV protection for HIV-negative women and a contraceptive, so a two-in-one product. Yes. Does this study have any implications for that type of research?
It might. I think we have to think about the multi-purpose prevention technologies, which we call that, and make sure that the two components in a multi-purpose prevention technology, say that 10 times fast, are both doing what they say they're signed up to do without having an interaction that causes one not to work effectively. So, you know, we're getting ready to start those. We've started those kinds of studies in early phase 1. We'll have to complete the pharmacology and make sure that it continues to look as good as we think it might.
Great. And so switching gears, I want to talk a little bit about the HOPE Act, which is a congressionally passed law that happened in 2013. Tell us about the HOPE Act.
So what the HOPE Act did was reverse a previous law that said you could not transplant organs from HIV positive people. And so what we have done by making it legal to transplant organs, we've given hope to, hence the name, to HIV positive people who are in need of an organ, because they are now eligible to have the kinds of transplants that would be from an HIV positive donor to an HIV positive recipient. And so if you think about one of the biggest challenges we face in transplantation, it's this waiting list. People get on the list and they wait 36, 48 months, even longer sometimes, and many people die on the transplant list because they haven't been able to get an organ. So for HIV positive people, this frees up a batch of organs that could be used specifically by HIV positive people under a protocol. We are studying this from the perspective of understanding if the organs last as long, if there are other things that we need to do in the context of HIV to make the organ transplant durable and maintainable as we move forward. So this is an important set of research questions, and we're excited by the work that's being done under the HOPE Act.
And so are people living with HIV more likely to be on an organ transplant waiting list or to need an organ transplant?
Slightly, but again, kidney disease, liver disease are all comorbidities that are associated with HIV infection, but hypertension that causes some of these is not unique to HIV, so I would say that there's a slight increase in the level on the recipient list going forward.
And so you described an HIV positive organ being transplanted into someone living with HIV. The law also contains provision for hepatitis C positive organ transplantations. Tell us a little bit about that.
So this is one of those really interesting biology experiments or findings. So we have a new class of antivirals that are not against HIV, they're against a virus called hepatitis C, which causes hepatitis C in the liver. So these drugs are so powerful that they're essentially curative. So under this act, you can take an HCV positive liver or kidney and transplant it into an individual that has no hepatitis C disease. The organ graft takes, you treat for 8 to 12 weeks, the organ and the hepatitis C is eliminated, and you are suddenly have a brand spanking new liver or kidney. And it's pretty spectacular that we can do that. And again, back to this waiting list issue, if a person is eligible or willing to take a hepatitis C, a liver that contains hepatitis C or a kidney, it can significantly reduce the amount of time they're on the waiting list. So, in talking to the investigators, there's consent and people sign up for these things. And the lead investigator, Christine Durant, talked about this today, that when talking to potential study participants who would be receiving these organs, they say it's a no-brainer. Why, once they know all the facts, why wouldn't they sign up to receive an organ like this? They're moved up the transplant list. It's a game changer for individuals.
That's great. And I think it's important to underscore, too, that these transplants are all taking place under the auspices of a clinical trial. So This is something that people have opted in for, whether we're talking about HIV positive to HIV positive transplants or hepatitis C positive to hepatitis C negative transplants, right?
Absolutely. That's an important point, is that, again, the purpose of why we're doing this research is so we truly understand the parameters of the transplant world and how this can improve what we're doing. It's also important for people out there, if you are interested and you're an HIV positive individual, you can get on the organ donor list. And I think that's another important activity that we would like to encourage is class participation on this. So if you are interested, go to the website organdonor.gov. I imagine it's showing up on the screen as we speak by magic. But at the same time, don't just get your name on the transplant list. Talk to your loved ones, talk to your family about that this is your wishes. It will make it so that if with your passing, you will be living on and you will be doing something good and giving back to another HIV positive person who is in desperate need of an organ.
And if you're just tuning in, I'm Anne Rancourt for HIV.gov, reporting from CROI 2018 in Boston, here with Dr. Carl Dieffenbach from the National Institutes of Health. And so switching gears again to HIV vaccines. So Carl, you were telling me about a really interesting presentation earlier today, a very early finding in monkeys, looking at gene profiling and how it might be used to speed up the flow of information about how a vaccine is working.
Sure.
Tell us a little bit about that.
So there's a group headed by a scientist named Louis Picker at Oregon Health Sciences Center, and he's working with a very unique type of HIV vaccine. It uses a virus called cytomegalovirus as the delivery vehicle, and he puts the monkey genes in. Again, this is all in monkeys, and we talked about the stage of research yesterday. So remember, this is early days. These are monkey experiments, but you'll see where this is going as I finish my story. So he puts the monkey genes in, and he infects the monkeys with this CMV vectored. Now it has the HIV genes in it. And the monkeys make very powerful immune responses. And then when he challenges those animals with the monkey equivalent of HIV, about half of them are protected from infection. And his work has always been right at the 50% level. About half the monkeys get infected, half don't. And it's really important then to try to figure out why. And so an investigator at the University of Washington named Michael Gale has a team that does these deep analyses of patterns of gene expression. What's going on in the body? You give a vaccine and all kinds of changes start occurring. And they can monitor what these changes are by looking at the patterns of gene expression. And they have been able to discover that within 3 days of giving the vaccine to these monkeys, there are very specific patterns of gene expression that get locked in, and they can, with 90% accuracy, tell me or you which animal would be protected and which animal would not. So what this does is two things. It allows us to sort and figure out what's going on. It also says we know the genes that we need to turn on to make sure that a vaccine works. So can we then figure out better strategies so we're not at 50%, but we're at 90 or 95% of turning on the right genes and not turning on the wrong genes. So this is a really exciting window into what we call the correlates or biomarkers of protection. It will work first in monkeys. Lewis thinks he will get his CMV human vector into people within 18 months. And so then we can use the same sort of immune profiling in people to see if we're going to see the same sort of patterns. So this is where the work gets replicated. And Lewis has done this with different routes of delivery. He also has a TB vaccine in the same platform that he will also be able to do this kind of analysis with Dr. Gale. And when we get into humans, we'll also be able to look at this. So it would be really good to build the database to make sure that we are confident that what we're seeing will work. This is the way science should work.
That's great. That's exciting.
It really is.
And so we haven't heard that much here at CROI about vaccine trials done in humans. Why is that?
So I think it's important that we go through phases in research, and right now, where we are on vaccines it's like we're cooks. Dinner's in the oven. We have 2 big clinical trials that are cooking. They're both in sub-Saharan Africa, a trial called 702, which is a direct repeat of the RV-144 protocol, but adapted to the Clade C or South African environment, and another study sponsored by Janssen Pharmaceuticals, but done in collaboration with the NIH and our partners, the HVTN, called 705, which is looking at a different system of achieving immunity based on an adeno-vector platform. So stay tuned. Both these trials are actively enrolling and should report out in a couple of years. So, like I said, soup is on. We'll see how it goes.
Great. And so we heard also earlier this week from one of our colleagues at the National Cancer Institute, Dr. Mary Kearney, about her work in the lab to look at HIV replication in lymph nodes, which are part of the immune system, while people are on antiretroviral therapy. Tell us a little bit about what she found.
So this is a really important study because if you think about the way we look at an individual on antiretroviral therapy, we measure the amount of virus in their blood. And often we say it's undetectable, and we all know that undetectable equals untransmittable. But there has been an ongoing debate is, well, it's not in the bloodstream, does that mean there could be, or some people think there could be, ongoing low-level virus replication in other anatomical sites, in places in the body. So the lymph nodes are a great place where lymphocytes, like the white blood cells that HIV infects, congregate. And so if you get a bunch of them together and a virus gets in there, they have a little bit of a party and the virus goes nuts and grows. But therapy is supposed to shut that off. That's what therapy does. It stops virus replication. So what Mary and her team have done is gone in and looked at the lymph nodes in people and have found no evidence of ongoing replication. And that is very important, simply because we think about U equals U. And we want to make sure that U, meaning undetectable, also means not just not transmittable, but also no ongoing replication. And her data is pretty definitive, and it continues to build the support for U equals U.
And so talk us through the concept of U equals U, excuse me, a little bit. Undetectable equals untransmittable. What does that actually mean?
So, we have spent a lot of time this week talking about the clinical trials process and the research process. And we've had a number of studies that have been completed between 2010 and 2016, 17, and 18 that actually looked at, does virus suppression, the ability to deliver the antivirals and suppress somebody to undetectable, what does that mean for transmission? And the landmark study of HPTN-052 demonstrated that virologic suppression gives you no transmission in a couple. There were 3 cases of transmission in that trial that were linked. In each case, they were also able to go back and show that those people were not suppressed. We also talked this week about the importance of replication. Can you repeat the study in a different population? And the partner study, headed by a physician named Roger, demonstrated that and this was also in MSM, there was no transmission in people who were between a HIV-negative person and somebody who was virologically suppressed. So that's what we mean by having a package of studies that are different enough, but are mutually reinforcing, and the data builds to a point where you are reasonably confident that you can take this to the bank, as it were, and that U equals U.
So these are large human clinical trials. That's what folks should be looking for.
Take years to accomplish, and again, but change public health policy. That's what we're looking for.
Wonderful. And we so appreciate everyone tuning in this week. It's been a great week here at CROI. We especially want to thank our viewers in Orlando yet again. Prospect, Kentucky, also extremely loyal viewer. Great to see you. Great to have you with us. Brooklyn, Maryland, and Gainesville, Florida. Hi, Mom. So thank you for watching. It's been a pleasure. Carl, thanks for joining me.
It's my pleasure.
For more information, please visit hiv.gov.